Today I'm going to talk about some of the work that we've done in the clinical microbiology division here at the UW, and you'll bear with me, some of the aficionados. I'm going to provide some introduction so that we're all on the same page. The UW is composed of a referral center that provides primary care in many different disciplines, and truly it's a center of excellence, and I think that together with our colleagues at Children's Hospital and the Fred Hutchinson Cancer Research Center, we together form a center that is truly outstanding with regard to clinical care. The breadth and depth of the care that's available here, I think, turns out to be both a microbiological liability, but also a microbiological opportunity, and we'll get back to this a little bit later in the talk. The goal here, of course, of all these different departments is to treat and cure patients. And to do that, they rely on making diagnoses, the determination of the nature of a disease, the Greek roots of deciding. It's a synonym with the Greek word diakrisis, which means dia meaning through and krisis meaning a judgment. And so when you or any other patient goes to see a physician, they formulate a hypothesis based on the history that you provide them, and they test that hypothesis in physical exam and reformulate the hypothesis and gather additional data using imaging and, importantly, of course, laboratory testing. And today this is laboratory medicine grand rounds, and in the Department of Laboratory Medicine we have many different divisions, all of which are charged with serving the diagnostic needs for this very large center. We serve as a reference lab for greater than 28% of the geographic United States. And today we're going to discuss specifically molecular diagnostics and their role in microbiology. So the mission really of that section of our laboratory is to support delivery of the best possible patient care by giving excellence in the science of diagnosing infectious diseases. And we have a large number of clients. If you pick a state and a large referral medical center, they've sent specimens to us because of the technology that our group has been able to develop. Now, they're all interested in the identification of the etiologic agents of infectious diseases because it's useful in patient treatment for determining the nature of disease, predicting the course and potential outcomes, tailoring the therapy, and very importantly, excluding non-infectious causes of the symptoms in the patient. One of the important rules of microbiology is that things you can't see can hurt you. And today we're going to talk about very small things and very large numbers. So the take-home for my presentation today is that rapid and sensitive capture of genomic information integrated with the traditional discipline of clinical microbiology enables robust diagnoses and accurate descriptions of new and emerging pathogens. This is largely a result of the diverse patient population that we serve. So, for accreditation, I'm required to announce what you'll be able to recite after we complete today's session. You'll be able to talk about the role and use of molecular technology and laboratory utilization, the efficiency or efficacy of the lab, and also a term that I'm going to call hospital fitness, which I predict may lead to some discussion. The bottom line, of course, is that this will result in improved patient care, and I would like to provide you with some tools that a large number of people have made a lot of contributions and allowed us to move forward. So the traditional approach in microbiology is that if an organism grows, it can be identified, and that allows us to do these things that we discussed: determine the nature of disease, a predictable outcome, how do you intervene and intervene effectively. One caveat is that if we depend upon growth, is that the turnaround time can be a function of the growth properties of the pathogen that we're interested in identifying. So the traditional approach has relied on signal amplification, and as you can see over here, there's a single bacterium. Now, no need to adjust your television set. It's actually very, very small. And in order for it to be large enough for us to see, we rely on signal amplification, which is sort of a tour de force of laboratory testing in all of the divisions. And we rely on a billion-fold amplification that will allow us to see a single colony, which contains about 10 to the 9th colony forming units. And visible to the naked eye, we can evaluate a number of different properties, phenotypic properties. And we're really going to attack basic biology in order to impact the diagnosis of infectious diseases. And so the first thing we're going to talk about is the flow of biological information in basically any cellular unit. So the phenotype of a microorganism, here you can see 3 different strains struck on blood agar, and you can see that there's clearing around each strain. This is diagnostically useful, and you can see that they cause the lysis of the red blood cells in the media. Similarly, a different organism can also cause lysis of red blood cells, but also have this metallic sheen. Now, these phenotypes result from the production of gene products. And gene products can be proteins, but they can also be described as antigens. So we can look at the phenotype, but we can also use antigens as an indicator of the presence of a microbe of interest. And so we can detect antigens, for example, using a common laboratory technique, latex agglutination, where latex beads coated with an antibody specifically recognize an antigen produced by a bacterium, leads to the agglutination of these latex particles when that antigen is present. But when it's absent, we don't get agglutination. Similarly, technology that's used all over the laboratory is that we can use solid phase antibodies to capture antigens and then use a secondary antibody coupled to an enzymatic component that allows us to detect the bound antigen. And again, here we can use amplification strategies in order to identify the organism that may have expressed this particular antigen. Now, gene products naturally are the product of the genotype. And so genes are responsible for the production of these proteins and antigens, which then are responsible for a phenotype that we can see on a plate. And so realistically, the genotype is really the blueprint for the phenotype that's manifest as gene products being expressed by a living organism. And then the phenotype is really the physical expression of that particular genotype. And so this provides multiple levels at which we can interrogate a microorganism to try to determine its identity, because that's going to influence how patient care is then subsequently delivered. So, how can we attack genotype? Well, this is a very nice slide that exemplifies the polymerase chain reaction, which I'm not going to go into any detail except to note that you can specifically amplify genotypic information. So if we have a particular gene of interest where there's 2 copies, when you make a copy, you get 4 copies, and when you copy 4 copies, you get 8. And 8 gives you 16, and 16 copied gives you 32. If you do this 35 times, you can end up with 68 billion copies. So that provides us with exponential amplification and allows us to effectively look for a particular genetic signature in a complex milieu. Now, when we've produced a product, as you can see here on this agarose electrophoresis or agarose electrophoretic separation, we can interrogate this particular product for its attributes that will then provide us with diagnostically useful information. So we can look at the length. We have capillary electrophoresis instrumentation that has single base pair resolution. We can directly sequence the amplicon. That's both accurate and becoming more rapid and less expensive every day. We can do melting curve analysis, which is a surrogate for sequencing, because the melting properties of a polymerase chain reaction product are dependent both upon the sequence of that product as well as its length. And finally, we can use nucleic acid probe hybridization, which is really a rapid and sensitive way of detecting signature sequences in that PCR product of interest. So the traditional approach, if it grows, it can usually be identified. And the caveat there is that the turnaround time is often a function of the growth properties of the organism. And so we're going to, I'm going today share with you some examples where poor growth properties of the organisms actually provide the opportunity for us to use molecular technology to aid in the diagnosis. So there are multiple levels of the flow of biological information that we can use for making a diagnosis. And in particular, we're going to focus when things don't grow well, it's difficult to find their phenotype. So we often rely on antigenic structures or on genotypic information. So an example of that, what is the overall goal here? The goal of the pathogen is to simply colonize your body, multiply, and then transmit. And really, as patients, what we feel is that when colonization or there's sufficient multiplication that you feel infected, today you might think you have a cold. A cold might feel the same as having the flu or having whooping cough. And after subsequent rounds of multiplication, the manifestation of the microorganism's attributes, genetic attributes, can lead to overt disease. Now, of course, the goal here is not to wait until we have diseases in order to treat them. We, of course, want to block transmission, but one of the goals is, with new technology, is to actually push back and interrupt this process earlier and earlier to manage quality outcomes for potentially infected hosts. So one example of that is Clostridium difficile. That's increasingly a cause of bacterial enteritis in the hospital. It has a substantial impact on infection control. Culture is slow and difficult. Therefore, you can use antigen-based detection schemes. For example, a bacterial antigen to ask the question whether or not C. difficile is present in the stool sample of the patient, and a separate reaction to look for toxin antigen, or the production of a toxin made by C. difficile that's responsible for the pathology in the GI tract. That's diagrammed here, where you can see a potential antigen that identifies the bacteria as well as the production of a toxin. Now, we just discussed the biological flow of information, and so the production of the toxin can also provide a phenotype or a bioassay, which is the ability of this toxin to kill fibroblasts, and I'm going to show you an example of that in a minute. The problem with this diagnostic strategy is that both of these assays suffer from a lack of sensitivity, and so this has been a long-standing issue that a number of people in our division have contributed very important ideas to the development of new technologies to improve our diagnoses. So when we look at antigen detection, the good news is that most patient specimens do not contain C. difficile, nor do they contain the production of the toxic antigen, and that's good news for almost 90% of the people. Unfortunately, for a small percentage, their stool samples contain the bacterium C. difficile, and those isolates also produce the toxin. And the critical issue here is that there's this interesting 10%, 9 or 10% of the patient samples where we detect C. difficile, and it's a question of whether or not those organisms also produce toxin. And so one question is, are these truly C. difficile that do not produce the toxin, or are they actually organisms that belong in this particular category? So one potential strategy here is for a patient sample to be delivered to the laboratory, perform this testing, and in the cases where it is clearly positive or clearly negative, the diagnostic algorithm is complete. And then to follow up on these samples with another test to ascertain whether or not they may be subpopulations of this particular group, and patients where treatment is important. Now, that seems like a reasonable approach, and so one follow-up is to use a bioassay to look for the presence of toxin, potentially in this 9% population where the result is equivocal. And so you can see here fibroblasts that are rounding up due to the toxic effects of the toxin, and that here are normal fibroblasts that are growing happily. So this would be a reasonable follow-up in that 9% of cases. When we looked at ordering practices, what we found to our surprise was that almost 40% of the samples that came to our lab, only antigen detection had been ordered. And then in another 61% of the cases, both the antigen detection strategies plus the phenotypic bioassay had also been ordered. And this suggested to us that there was an opportunity here to have an impact with other technologies. And so I'm going to make a long story short, but the approach is to specifically amplify genotypic information for the gene that encodes the toxin, the TCDB gene. And the targets here are one to identify the bacterium and one to ask the question whether or not the bacterium contained the gene that produced the toxin. And at least one version of this test has been validated on stool samples, where in fact we've shown that the genotype in fact accurately reflects that a gene product is made, i.e. there's an antigen that we can detect, and that this gene product leads to a phenotype, which is, in the bioassay, it provides cytotoxicity. So then when we go back and use PCR to evaluate the antigen detection system, what we actually found is in this critical population of indeterminate results that at least half of these stool samples were in fact truly contained toxigenic C. difficile. So what are the impacts of this what are the impacts of this technology for diagnosis? And that is, if we go back and consider ordering practices, if you only order the antigen test, I just showed you that you're potentially going to miss half the cases. When you order both tests simultaneously, 90% of the time the second test is not required. So a reflexive strategy where 90% of the specimens are only going to require the antigen test. In 10% of the cases, that is where we detect a bacterial antigen, but in the absence of toxin, we can use PCR. And that PCR is sensitive for detecting the toxigenic C. difficile. So what's another example of application of molecular technology? Many of our services support compromised patient populations that have reduced defense mechanisms because they're being treated for cancer or getting some sort of transplant. And infection is an overwhelming cause of morbidity and mortality in these patients. And importantly, we invest large amounts of our healthcare dollars into these particular patients. Very typically, 10 to 15% of inpatients in a referral center like ours can provide up to 40 to 50% of the revenue of a particular hospital. So for these patients, invasive aspergillosis is a big problem. It has extremely high mortality. Diagnosis is difficult, and mysteriously, this organism defies cultivation. So there's a two-step approach to identifying this, which is to amplify a target sequence and then use a hybridization probe to detect a signature sequence in that product. And so hybridization works when two probes like this hybridize on a target sequence such that we can use fluorescence resonance energy transfer to detect the specific hybridization of these probes to a particular amplified signature sequence. And a typical data looks like this, where we get a melting curve and detected by fluorescence. And this particular system is very sensitive, detecting less than one genome of DNA in extracted samples. So with the help of Brigitte Lemay, we applied this to a patient population, a stem cell transplant, and we compared it with an antigen test, and you can see that basically there's three categories of diagnostic finding: that the antigen, both PCR are positive, that the antigen is positive and PCR is negative, or that the antigen is negative and PCR is positive, in both proven and probable cases. And what you can see here is that about a third of the cases were positive for antigen and PCR, and about a third of the cases were positive for antigen only, and a third of the cases were positive for PCR only. So the addition of PCR increases the diagnostic yield in this strategy by 50%. So we've talked a little bit about patient care and the route to finding diagnoses, and we're going to talk now more about the cost to make diagnoses and laboratory efficiency. So one example is the non-fermenting gram-negative rods that we find in the sputum of cystic fibrosis patients, which are clinically significant because the current thought is that any organism present in large quantities for a chronic period of time stimulate inflammatory responses, which are part of the disease process. they frequently turn out to be difficult to identify because of phenotypic variability. They become less reactive or inactive with time, and that's largely due, interestingly, to mutational events. So work presented that was published in PNAS that came from Maynard Olson's group, they actually did whole genome sequencing of Pseudomonas aeruginosa isolates from a patient at 6 months of age and followed that patient through time to 96 months of age. And you can see that during that time, the strains acquired 68 mutations, losing serotype antigenic specificity, altered LPS biosynthesis, altered secretory systems for delivering potent virulence determinants, motility, toxin production, drug efflux. There was a gradual increase in antibiotic resistance in this population as well. So for that reason, this group of particularly clinically important non-fermentative gram-negative bacteria, as well as Staph aureus, all present with phenotypic variability that completely defy characterization using traditional methods. And so the hypothesis, and this is largely the work of Nancy Goodyear, that was that an algorithm that can combine both phenotypic and genotypic approaches, i.e. in this case DNA sequencing, could decrease turnaround time, increase the accuracy of identification, and then decrease the overall cost, particularly if you consider the cost in delaying the identification and considering the clinical significance potentially of some of these pathogens, not just for the therapy of the patient, but also for the types of therapy that are available to the patients. So we looked at 98 consecutive specimens from CF patients and categorized the work that went into a phenotypic workup and considered cost analysis and error rate. And I could devote an entire grand rounds to target selection, but I think the important point here is that we look for species-specific DNA sequences indicated here as orange, pink, and burgundy bars, surrounded by green conserved primer binding sites so that we could specifically determine which species was present. And what we found is that phenotypic identification frequently works well, and we get about 85% of the organisms correct. That's 15% shy of perfection, which is our goal. So we came up with an integrated identification scheme, which I hope you won't try to memorize at this time. The important point is that we've worked very hard to come up with algorithms that provide efficient and accurate diagnoses. And so the take-home of this study was that using an integrated approach, the error rate was reduced to 0, the turnaround time was reduced, the cost actually increased 5% per sample, but that has to be taken into consideration that we now have a much higher rate of accuracy and a decreased turnaround time. It's potentially easier to apply molecular technologies when the organisms are absolutely positively impossible to identify. So we frequently get very, very difficult isolates to identify from outside hospitals. So in this study that was largely conducted by Brandy Limbago and other members in the clinical lab, we looked at 100 consecutive isolates from 19 outside hospitals. And here the hypothesis again was that we could integrate phenotypic and genetic approaches to get an identification that would be both accurate and cost effective. These are problem bugs. They've received extensive workup already. They're refractory to traditional methods. And the notion is that we could increase accuracy, reduce turnaround time, and do it at reduced cost by integrating the two. We were wrong. Actually, integrating the two is more expensive. The cheaper approach is actually to go directly to a genotypic method, and that results in a cost savings of almost 20%. So the accuracy increased from 32% to about 100%. The turnaround time was reduced, and we could also reduce the cost by 20% using a strictly genetic approach. So now I'd like to talk a little bit about hospital fitness. And the things that I think where molecular technology can provide additional information that helps a hospital run effectively, and these are very difficult issues to measure in terms of quantitative cost savings and impact, but the places where they've had impact since I've been here are in the infection control and regulating and developing both policy and procedure for trying to limit this nosocomial infection in our hospital and for other hospitals that use our services. The use and need of patient isolation, the appropriate utilization of antimicrobial agents, and effectively the sooner hypothesis for effective medical care. The sooner we identify your problem, the sooner we can fix it, the sooner you go home. I think that's beneficial for the patients. I think that's beneficial for healthcare FTE. So finally, the technological advantages of using molecular methods are that problems that are previously not identifiable can now be identified. So specifically what I'm talking about here is how do we exclude non-infectious cause of symptoms? Or in other situations, how do we exclude the possibility that infections aren't a cause of the patient's problem? So this has led to a very rewarding opportunity to help clinicians, not just at our hospital, but at other outlying regions with difficult cases. And so part of the reason this is so important, I think, is just to give a plug to some basic science investigation. There's a Center for Excellence in Genomic Research here that's headed by Mary Lidstrom, who's the Vice Provost for Research here. Our other collaborators are Babak Parviz in Electrical Engineering, Lloyd Burgess and Norm DeVicki in the Department of Chemistry, and also Dee Dee Meldrum, who's the PI for this center, who is the Dean for the School of Engineering at Arizona State. And basically the process whereby normal cells undergo cell death and lead to inflammation is mechanistically similar in a diverse number of human conditions, which include malignancy and heart attack and stroke, and as well as a large number of infections. So at a mechanistic basic level, there is a reasonable understanding for why cancer and infection might be confused. So we're all aware of the great imitators. Karen LaFay, for example, would appropriately point out Histoplasma capsulatum, and Carolyn Wallace would indicate Mycobacterium tuberculosis. Great imitators, well known in medicine, to imitate other diseases. So there was a 54-year-old individual with a mediastinal mass who was actually sent to the oncology clinic with a working diagnosis of lymphoma. I think that individual was pleased to receive the identification of Bartonella as a cause and received antimicrobial therapy as opposed to chemotherapy. Another example is a 39-year-old individual with a CNS mass that's suspicious of malignancy, and no organisms are seen on the cerebral spinal fluid, nothing grows, but we provide them with an identification of mycobacterial infection. And similarly, there are other problematic mysteries that are faced by the clinicians on a regular basis where molecular technology can afford them with some additional tools. Specifically, this is a GMS image of a lymph node biopsy from a young person with AML. And while as a microbiologist I find it extremely beautiful to look at, the question is it yeast, or is it a mold, or is it an extremely funky bacterium, or is it simply a contaminant? And it turns out it's probably Pseudomycelium, and we identified it as Candida albicans. Similarly, a 23-year-old individual with refractory seizures undergoes open craniotomy, where biopsy reveals no evidence of malignancy, no bacterial fungal organism seen, but the inflammatory process is consistent with infection. It's culture negative, and all the serologies for infectious agents were interestingly negative. We identified Tinea solium, which is cestode. This is basically a pork tapeworm. And so this is not just an anecdotal case. This has happened more than once. And so what was it that allowed us to do these things? We've developed a platform where you can amplify species-specific DNA because it's flanked by conserved primer binding sites. And the information derived from there has largely been based on the hard work of the people in the clinical labs here to allow us to ascertain that it's phylogenetically informative, that is, two bacteria are more closely related to each other than they are to a fungus, than they are to cestodes, and that it's also taxonomically accurate, which means that we can reproducibly identify organism X in a variety of different settings and categorize it the same way. And the accuracy of this method is enhanced by multi-locus analysis. That is, we looked at multiple sites in the genome, and that enhances the sensitivity of this method. So again, the targets were to use these species-specific DNA sequences in orange, pink, and burgundy, flanked by regions of conservation, so that we could amplify these specifically to ascertain the identity of an organism. And the success of this program is based on phenotypically validated database. And that's a result of the hard work of the people in the clinical lab, so that we can genetically characterize well-characterized strains and come up with a large number of diagnostic alleles. That then positions us, provides us with a foundation for interrogating the specimen that's culture negative, that shows presence of inflammation or the presence of microorganisms, but where other testing, such as cultures, are negative. We can apply these to other laboratory samples, which include blood culture, directly from tissue specimens, from paraffin-embedded formalin-fixed tissue, and this is where, in particular, for reasons that aren't completely clear to us, that multi-locus analysis has dramatically increased sensitivity. So from this, I conclude that if it grows, it can usually be identified, and there are many reasons that we would like to identify a microorganism, that we have to modify this a bit to emphasize if it grows, it can usually be identified. So the practice of clinical microbiology is a very diverse process. It's beautiful. It's beautiful to look at, but it's mind-numbingly complex. And so some important pieces of information to think about as we go forward in the future. And we talked for a few minutes about what the content of a dream machine might be. Carl Woese in 1996, he predicts that greater than 99% of the microbial species defy cultivation. Hawksworth predicts that there's over 1.5 million species of fungi on our planet. And our very own Paul Swanson, who's head of hospital pathology, thinks that it's reasonable for an anatomic pathologist to potentially be able to identify 1,000 entities. If you multiply phenotypic variability, which we've talked about this afternoon, times the number of new niches that are being formed for these pathogens, and largely that's a result of the success of all the centers or the success of all the services here at our medical school. As their medical technology progresses, they're providing new patients with a new set of medical problems that have been solved that provide a new environment for additional pathogens to grow. And so this really is a bit of tyranny. It's a tyranny of numbers. And so there's the increasing number of potential pathogens due to these new niches. There is a human limitation for our ability as individuals to account for the large number of pathogenic entities. And even if we are capable of identifying all of them or knowing all of them, that's compounded by variable phenotypes. And furthermore, that in potentially having a variable phenotype is the best case scenario. They may completely defy cultivation. And so this is really like the microbiological equivalent of the need for the integrated circuit. That might predate some of the members of our audience. But when we went from tubes to silicon, that was a dramatic increase in our capabilities. So considered in the context of a center where we're trying to cure patients' diseases and we're doing our best to bring in new technologies that are going to provide a new patient population that are going to be susceptible to a new set of pathogens, the responsibility of running a clinical microbiology lab seems rather large. Responsibility seems rather heavy with respect to really providing the kind of service you would like for you or someone you love as a patient. So the question is, what should we do? So at a forum like this, you should never ask a question unless you have the answer. And so I searched for the answer, and I found it. So I don't know, for the Trekkies in the audience, you might recall that in a cargo bay, Worf actually had an accident and broke his spine in many places. And Beverly Crusher was actually able to recapitulate and reform in vitro a brand new spine for Worf by sequencing the genome at 10 to the 9th base pairs per second. So in the dream machine that is coming potentially down the road to help serve the diagnostic needs of the clinical microbiology lab, we don't have the capacity, nor do I think we'll get to 10 to the 9th base pairs per second. That means you could do the whole human genome in about 3.2 seconds. But we're moving in that direction in a positive way that I think is going to have a dramatic impact on the way clinical microbiology is practiced in the future. One agenda that is currently part of the National Institute of Health is to develop personalized genome sequencing. Now, most of what I've told you today actually is a direct product of, or a direct benefit of, utilizing technology that was primarily developed for the Human Genome Project. And so, as goes the National Human Genome Research Institute, microbiology will benefit in a great way. So, I had a conversation with a couple of people, one of whom is Jeff Schloss, who's the program officer, our program officer at NHGRI. And between 2002 and 2005, the costs per base of DNA sequencing have decreased by 70%. And Mark Geyer, who's the head of extramural research at NHGRI, tells me that the NIH is currently investing about $25 million a year to developing sequencing technology, with the outright goal of, by 2014, decreasing the cost to do DNA sequencing by 10,000-fold. As quoted in Science, George Church, who's just a phenomenal scientist at Harvard University and has made some very remarkable contributions to sequencing technology, he said that there is no physical principle that says we shouldn't be able to do a $1,000 human genome. So what does that mean in the near future coming to you? Who is Future Lab? There's 3,200 megabases in the human genome, and if we're going to be able to do that for $1,000, I'd like to do some simple math that I find very, very exciting. The 35 megabase genome of aspergillosis, we'd be able to do for about $10.94. The 15 megabase yeast genome, we're going to be able to do for about $5. And we're going to be able to do individual bacterial genomes for $1.50. Now, that will provide us with a great deal of opportunity. I would say it's going to, the relationship between traditional microbiology and molecular methods is going to continue to grow. And I think with regard to in vitro resistance detection, the ability to sequence entire genomes is going to position us to make rapid and accurate identifications of antibiotic susceptibility and resistance. We're still going to need a robust database of viable organisms upon which to base this, but in many situations we're going to be able to skate quickly to the answer using whole genome-based information. We're also going to develop and understand more about microbial communities in health and disease. So the normal microbiota, the microbes that share your body right now, in fact, the microbes that you share space with, since microbes actually outnumber your cells on a per cell basis by about 10-fold, are very important in maintaining our health. And it's likely that the application of probiotics to modify our microbiota, as well as our ability to assess the host's ability and the microbial communities present in a host, will allow us to predict susceptibility and resistance to other diseases. And going a little bit further out on a limb, in collaboration with my colleagues in the immunology lab, I would propose that together with the high throughput assessment of host responses and the assessment of the capacity of hosts to respond, that we're going to have unparalleled therapeutic and diagnostic capabilities in the decade to come that are truly going to transform the favorable outcomes for infected hosts. The take-home today is that rapid and sensitive capture of genomic information can be integrated with traditional microbiology to assist in our ability to diagnose infectious diseases. And now I would like to spend a few minutes and acknowledge the important contributions of many individuals. The entire foundation for the molecular diagnosis lab is built upon the hard work of the clinical microbiology staff at Harborview and the UW. They provide effectively the database of phenotypically well-characterized organisms from which we can gather genetic information to build our testing algorithms. In molecular diagnosis, many of these people are on a rotating basis or have served and now gone on to bigger and better things. But I would like to particularly point out some important contributions. In particular, I mentioned Nancy Goodyear's work with the cystic fibrosis isolates. I'd also like to mention Sarah Rasulian-Barrett, whose contributions to fungal DNA extraction allowed us to get our first NIH grant to develop these technologies. And very importantly, I'd like to recognize the contributions of Jenny Prentice, who did a master's degree studying molecular identification and the speciation of mycobacterial isolates. And there's a significant jump between performing a test on a research basis and being able to provide it as a clinical service. And Jenny had a very large hand in making that happen. We also have, I have to thank the technologists with expertise in mycology and mycobacteriology. And then I would like to also acknowledge the courage and spirit of the unofficial informatics unit. Laurel, on her own time, helped get our initial website off the ground that I think led to some of us receiving some of the more interesting cases. And two really outstanding residents, Noah Hoffman and Andy Hufnagel, have had a great deal of influence on Big Dan. And Big Dan, as Dan Hugestrad, has played large. I didn't get to talk about any of his work today in allowing us to do sequence analysis in a high throughput fashion. The current clinical fellows, Amanda Harrington and Jean Miguel Balada-Lasat, continue to play an important role in evaluating clinical specimens that come into our lab. And there are a number of other people that I would like to thank, principally Ajit Lemay and his research group for their help with the aspergillus detection schemes, and to Ajit for his critical contributions and feedback. I'd also like to thank a host of collaborators and contributors, in particular, that have provided us with a great deal of intellectual stimulation and terrific ideas, and in particular, Farrick, Ann, Tom, and Jen with regard to C. difficile diagnosis. And then one of the aspects that I think has been quite rewarding for us as an educational institution is a lot of our former employees now populate other sites where they've learned a lot about molecular technology at the University of Washington, and now they're teaching other people. And some of them have gone on to medical school, and some of them are at the CDC, and many of them direct their own labs and public health labs in cities throughout the country. And so with that, I would like to thank you all for your attention and take any questions that you might have.
We have plenty of time for questions. I would just ask, Brad, that you would repeat the question that's asked so we can all hear.
Doctor?
First of all, that was an excellent talk. Thank you very much. Second of all, it wasn't Dr. Beverly Crusher who sequenced at 10 to 9 base pairs per second. It was, in fact, the other doctor, played by Diana Muldaur, who substituted for Gates McFadden briefly. Wayne, do you recall the name of the doctor? Anyway, so that was who did the sequencing.
So,
but the question, I have a question.
okay.
The question is this. It gets back to this. It's an ancient microbiology problem, and that is, how do you distinguish actual pathological infection from just colonization by an organism that you can pick up by PCR and sequencing?
That's an outstanding question. And I think that's the big bugaboo. If you go back to the slide where I talked about colonization, multiplication, and subsequent the transmission of the organism, which in that same time frame, the host goes from knowing nothing at all at initial colonization to having some physiologic response to bacterial replication, for example, let's say, that gives an indication that you're infected, and then followed by additional rounds of replication that lead to the formation of disease. And so the question, a good question is, how do you know it's a pathogen? And that's where you have to rely on other laboratory data, other clinical data, additional imaging, et cetera, in order to make that assessment. Now, over time, I think we're going to be able to ascertain where colonization, where and how colonization may lead to pathogenic events. We all know for certain that before you have disease, the organisms have to be present. So although it is a dogmatic sort of fragmentation of the process, with time, I think our abilities to be able to identify what will lead to infection will grow. But that remains a primary issue to be solved. Now, I would also add that in immunocompromised patients with the appropriate additional data, the question actually could be reversed and say, how do you know it's not a pathogen? And so one of the points where I think molecular technology plays a useful role is in patients that are susceptible to completely new entities. Now, there's almost always resistance and reluctance on the part of myself, our staff, and all the physicians we interact with to identify an organism for the first time as being a pathogenic entity. So I would just share with you one anecdotal experience, which was when I was a resident and over at Harborview Medical Center, Ann Larson and I identified a pathogen that, of course, no one thought was truly a pathogen. But that was back in 1994, and subsequently there have been many case reports, and now it's well established to be a pathogen. So at some level, part of that game, Dr. Sabbath, is the courage to be first, and that means you might be wrong. But we're hoping that we'll be right more often than we're wrong. Dr. Fong?
So I enjoyed your talk very much, Brad, and your lab has really, I think, pioneered the replacement in many cases of slow, difficult phenotypic methods with genotypic methods for identification. Another area in the lab, though, that we sometimes have to rely on difficult or unreliable phenotypic methods is for resistance. What do you think is the potential for replacing some of these phenotypic resistance methods in bacteria or fungi with genotypic analyses?
You repeat the question for our teleconference.
I will repeat the question. That's a really outstanding question. Dr. Fong asked what the capacity is for genotypic methods to have an impact in the area of ascertaining antimicrobial resistance. And the reason it's a wonderful question is you could also consider it a loaded question. But I think the primary shortcoming, so there's been a lot I would call almost hoo-ha published in the literature about the use of some genetic methods to attack this problem. Now, there are definitive examples where a particular acquisition of a transposable element or a particular signature sequence, to use the universal language that I developed for the talk today, that you can reliably predict is going to yield a resistance or susceptible phenotype. The bigger problem is, though, is that these are haploid organisms with very rapid generation times, and they evolve at a very high rate. And so the ability to have a specifically targeted genetic test to a particular element over time is going to have an extremely small or limited utility over time. What changes the dynamic in a significant way is not looking in a targeted way, but to do it in a genetic approach where we're simply going to look for what are the equivalent of single nucleotide polymorphisms or genetic traits that are connected that provide a genetic signature for a resistant organism. So this is going to be the task of informatics would be my answer. But the ability to capture a picture of the entire genome and make an association with potential phenotypes, I think that holds promise. Of course, it is yet to be ascertained, but I can envision where it would have an impact that we can take when we have whole genome level genetic information, that we're then going to be able to impact antimicrobial resistance detection. So I'm hoping that potentially in the next 10 years that will be possible in selected cases. And I think, as you mentioned, the place where we would start are difficult to grow organisms, or organisms where the methodology for predicting in vitro resistance those methods are less reliable.
The genetic code is very diverse, and for example, I remember reading one time that any human being's identity can be unambiguously assigned by only looking at like 14 or 15 different SNPs, because the degeneracy of just 14 core base choices is just enormous. Given that, why the push towards sequencing an entire genome when there's going to be so much information and only looking at probably only 20 different places in the genome, and probably 20 genes have to have such an enormous amount of information? How much more are you going to get over sequencing every single one of the 3 megabases?
Okay, so the question is, why is it that whole genome sequencing may provide us with additional pieces of information? And I would agree with you, Dr. Baird, that with respect to identity, it's not going to be required. And so one way I tried to illustrate that was with some of these mysterious cases where we were able to provide identification that fit with the clinical picture. So I think for identification, I agree with you. But for other complex traits, like the point that Dr. Fong brought up with in vitro resistance detection, that's where I would say that we do not have sufficient knowledge, and I'm not confident that looking at maybe 14 or 15 different loci is going to provide us with sufficient information. Now, I could be wrong, and if I am, that I won't be crying, and that's because the technology will reach that threshold before it will reach whole genome threshold. But that's the primary area where I think it's going to have a great impact.